Assalamualaikum warahmatullahi wabarakatuh. First of all, let me introduce myself. I'm Ima Rafiana from Department of Agrotechnology, Faculty of Agriculture, 11 Maret University. On this opportunity, I will present my research that titled The Effect of Parental Plant and Washing Materials on Seed Germination of Second Generation Apple Cactus Cereus spp. I will start my presentation with the introduction of my research. Cereus belongs to the neotropical columnar cactus. Some species that have been found are Cereus peruvianus and Cereus jamakaru. In this seed of apple cactus, there is mucilage that becomes a factor inhibiting seed germination. Seed washing treatment before germination can be one way to remove mucilage and increase seed germination. In this study, used the seeds of the second generation of apple cactus. Second generation seeds generally have some differences from their parent. Therefore, research needs to be done to find out the effect of parental plants and washing materials on the germination of seeds of the second generation apple cactus Cereus spp. This study used a randomized complete block design or RCBD using 3 parental plants and 5 washing materials with 3 repetition. The resulting data was then analyzed with ANOVA 5% and continued with the MRT 5%. The observed variables were seed viability indicated by maximum growth potential, germination percentage, and mean germination time, and also from seed vigor that indicated by germination uniformity, germination speed, and vigor index. Next is result and discussion, general research condition. First, before sowing, the fruit is first stored in the refrigerator at a temperature of 3 degrees Celsius for 2 until 4 weeks after the fruit is harvested. The seeds used are derived from the ripe fruit of second generation of apple cactus. Ripe fruit is indicated by the skin of the fruit has been red, reddish yellow, or reddish green. The seeds to be sowed are selected with criteria such as clean and healthy seeds. Next is characteristic of apple cactus seeds. The first image is the apple cactus seeds that have characteristic is black color 2 until 4 millimeters and has obovate shaped. The second image is the normal sprout of apple cactus seed that consists radicle, hypocotyl, and cotyledon. And the third image is the abnormal sprout. Next is seed dormancy. We can see from the diagram the highest number of hard seeds that appear compared to the number of normal, abnormal, and dead seeds. This can be happen because seed storage in low and not constant temperature and cause the seed skin to become hard and resistant to water and oxygen. Next is seed viability indicated by maximum growth potential. At 5% ANOVA results, parent plant and washing materials have no significant effect on maximum growth potential. The maximum growing potential is only ranges from 17 until 39%. Next is the seed viability that indicated by germination percentage and mean germination time. At 5% ANOVA result, parent plant has significant effect on germination percentage and mean germination time. Based on the MRT 5% result, show that the natural crosses of long spines has the highest germination percentage, 41.47%. Meanwhile, the long spines and short spines has the shortest mean germination time is 16.98 days. The result related to the use of female parent plants on crosses. Long spines when used as the female parent produce better germination than short spines as the female parent. The result are following the finding of Pulungan et al., which states that maternal affect seed germination. We can see that the seed viability is very low. According to Kartasaputra, seeds with hard skin can cause mechanical resistance so that seed embryos cannot appear to grow as sprouts. Next is seed vigor. Seed vigor that indicated by germination uniformity and germination speed. From 5% ANOVA result, parental plants and washing material doesn't give a significant influence or significant effect for germination uniformity and germination speed. From the diagram and the average tables, the highest average is only at 14.93% for germination uniformity, and from the germination speed is only at the value of 3 until 31% of ethanol. Uh, in this study, based on the seed chart, the speed of seed growth that represents a strong seed failure when it is worth more than 30%, while the seeds with germination speed less than 30% belong to the failure of seed is less strong. Fabriani and Widajati stated that the declining value of germination speed may indicate a decrease in the quality of seed failure. Next is seed failure indicated by failure index. So from the 5% ANOVA, the parent plants have a significant effect on the failure index. This is supported by the result of the previous research. So this research is we have the natural causes of long spines has the highest failure index, as well as long spines and short spines. From Julindra, who also obtained the result that the use of Cereus Jamakaru or long spines as a female parent produces with better quality. Sutopo, namely the low failure in seeds, can be caused by several factors such as genetic, physiological, morphological, cytologic, and others. In this study, it is suspected that the storage of fruit and seeds of apple cactus, as well as the use of fruit at an unequal age, led to low germination of seeds. Next is seed washing treatment. Barrios et al. States that the characteristic of cactus seeds from 40 species indicate the presence of a layer of a mucilage enveloping the seed testa. The mucilage that envelops the apple cactus seeds contains glucose, fructose, and organic acid. Because washing treatment has no significant effect on all variables, here is average table so we can see the result. Washing with pH 9 has the best germination than washing treatment with other materials, in line with the statement of Luz et al., which states that the removed mucilage in the cactus fruit required immersion treatment in substance with alkaline pH, in example, sodium hypochlorite 10%. Next, we can see from the image, the washing treatment using pH 9 can separate the seed and the mucilage. So the extraction process is more easily rather than the other washing treatment using the other material, because uh, it can minimize mucilage that envelopes the seed. This is in accordance with the result of Purwanti, which states that the pH 9 or alkaline can clean the seeds of dragon fruit from mucilage that envelopes it. Next is conclusion. Based on the results of the research that has been obtained, it can be drawn conclusion as follows. First, parental plants have a significant effect on germination percentage, mean germination time, and failure index. The use of long spines as female parents results in germination better than the short spines as female parents. Second, washing treatment is using a variety of materials has no significant effect on seed germination, but washing with pH 9 can facilitate the extraction process more easily. Third, there is no interaction between the parental plant and the washing materials treatment against the germination of the second generation of seed apple cactus. Next is the references that I use for the presentation. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.